For those fish that encountered Site 9 when OMR was greater than minus 4,000, much more negative reverse flows, 39% of the fish detected at Site 9 were detected at Site 11, suggesting that there was no response to OMR in terms of this particular migration pathway for these yearling steelhead. And with that, I'm going to let Steve.
Dr. Hansen, before you pass the baton off, I mean, you talked about transit times from above Red Bluff on the Sacramento system through the Delta and the lack of response to flow. Is that carried forward into saying that there is no positive effect either on up migration or out migration from pulse flows to try and stimulate either up or out migration? No, because the transit times that I was looking at were basically a 30 or a 60-day average or some longer period of averaging. We've seen that short-term events, storm events that create turbidity, increase flows, are important as migration cues for juvenile salmon moving out of the system. They're also important for adults migrating upstream in terms of providing attraction flows and an upstream stimulus for migration. So that is not covered in terms of, or wouldn't be detected in terms of the types of analysis that I was doing. As we start moving more into the application of acoustic tagging, we're going to be able to really watch how these fish move in response to pulse flows and turbidity events, those kind of fine-grained reactions that we simply now only take, rough estimates of. When you do that analysis, will you compare the results from a naturally occurring storm event and the turbidity that would go along with it compared to a man-made pulse release, if you will, to provide the same effect? Would you expect to be able to see a difference in the efficacy between the two events that were designed to mimic, the man-made one designed to mimic the naturally occurring?
I think we will be able to do those kinds of comparisons. And in fact, some of that work is started experimentally on the McKelmy and the Stanislaus and other rivers where short duration pulse flows are now being tested as a result of reservoir releases to see how they stimulate downstream migration. And what we're finding are mixed results. In some cases, we see good response. In many cases, we don't see a good response. So as we start looking at these natural events, we'll be able to better separate out the effect of the flow change versus the turbidity change.
Thank you.
Yes, thanks for that. I would add a third question, which is both at the same time. That is, so there's 3 data sets. There's actually pulse releases during storm events from reservoirs. That's a question I have, in addition to them separately.
Yeah, and from a management strategy, we've in various river systems looked at 2 kind of scenarios similar to what you're describing. We've looked at augmenting a pulse that occurs so that you've got synchrony within the watershed. And then we've also looked at adding reservoir flows after the natural pulse occurs so that we've got a longer duration. And so those kinds of studies are being conducted. We're learning a lot through that process.
Mr. Chairman and board members, I'm Steve Kramer, and you've heard from Dr. Hansen a variety of factors to consider, and you've heard that from some other presenters prior. My task here is to talk about some of the challenges that you will face and some thoughts about how to overcome them when you try to integrate all of this best available science. There are a myriad of variables to consider, and so the pathway forward is not always obvious. So here's just a list of ideas I want to touch on. First, the challenge of how you would account for all of the variables that have been addressed. Second, how would you discern which ones matter most? And then finally, how do you then discover the balanced solutions that could answer a number of competing needs? So first of all, I'm going to talk on that first one, how to account for all of the variables and the importance of doing that. This first, I'm going to use 2 slides to illustrate this. This first one is an example to kind of set the problem up. This is a graph of the abundance of Chinook these would be primarily fall Chinook produced by the Sacramento system, actually really the Central Valley. But what you have here on the y-axis is the Sacramento index. It's used in fishery management, so it's compiled each year. That's the number of fall Chinook that were spawned, the black bars, and the number that actually were caught in the ocean before they got to the river. That's in the gray. And then the number that got caught in the river is in the lighter portion here. A couple of things about this graph. Starts in '83 and extends all the way out to present. First of all, you see really high variability between years. That characterizes the populations of Chinook up and down the west coast, even in Alaska. There's nowhere where you can depend on them to always be the same. They vary substantially between years. Another thing to note here, you can see a lot of the total abundance is accounted for in the harvest that is taken. And then finally, what I'm going to use for this illustration here is out in 2008, 2009, we saw a complete stock collapse. There was no fishing allowed, and we got very few spawners back. Why did that occur? And that is the kind of the question before any challenge to find regulatory relief to a problem. Why did this happen? Let's see. Scroll down. All right, let's move to that one. Okay, so this is an example of how we address that problem. This was actually a graph and an analysis that was done by the National Marine Fisheries Service. It illustrates walking carefully through the life history to figure out where the problem occurred. First of all, if you go up to this line that's drawn across, it's drawn at a level that it represents the average over a long term. So each of these factors for those 2 years, and these are brood years 2004, 2005, those are the broods that resulted in the adults at the time of that stock collapse. So brood year is the year the parents spawned, and the kids are the ones you track forward. In 2008 and 9 didn't return. So in these 2 years, we can see the number of spawners was above average, or right about average. The number of hatchery releases, so did we let those go? Yes, the normal number there. How about the number of smolts, which would be the catch per unit effort at Chips Island? So that's the measure we use for the abundance of smolts leaving the system, and that would be natural production, and it was actually slightly above normal. And then the next signal we get is at age 2, so this is estuary to age 2. In estuary, we know they were Feather River hatchery fish. We have an estimate how many arrived at the estuary, how many then moved out till age 2. That's when they start appearing in the ocean catch. And there's your first signal that we had a dramatic decline far below normal. There's where the collapse occurred, and it continues to show up as you move to older ages. A whole lot of other data showed that as well to confirm that many organisms were affected. It was a problem in the ocean. But this figure illustrates the importance of going through a sequence of the probable causes to identify which was the actual cause of the outcome that you're observed and would like to alter. One point additionally, the ocean harvest is another one of those high variables that we do have control over management capability and is a big factor in determining the run sizes we observe in the Sacramento Basin or the San Joaquin. So for a number of years, it's run 80 to 90%. That was through the 90s. Then along came the Endangered Species Act. There was reductions specifically to allow endangered stocks to recover. And at what is being offered under that circumstance now is still 50 to 60% harvest rates. Even when they closed down the whole fishery here out in 2008 and 2009 along the coasts from California to Oregon and even into Washington, they quickly have restored to harvest, as many would think this is a substantial human benefit, but at any rate, it takes a number of fish, and now it's already back up to 40%, immediately following recovery of that one decline. So you see substantial variability and very large effect that is related to harvest. How do you then accumulate these and move to that idea of which one matters most and what would be a balanced solution for all the needs that compete for the water and for the fish? This is a life cycle modeling tool that enables you to keep track and account of all of those various factors. First, we start at the core that just represents the life cycle. And here you can see those different stages are in different places. Some are in the ocean, some are in fresh water, some are high in the system. As they smolt, they're lower in the system. Now, as we move to the next core, you see that the survival that connects one to the next is quite different at different life stages. In some life stages, it's real variable, and it's related to density and other factors. It's quite low in what happens once they leave and they go to the ocean. There's also a real difference in the proportion that mature at each age. All of those things influence how many fish you get back today and how you would interpret what went wrong or what went right about the run you have before you.
Steve, I asked a question yesterday that either wasn't explained well or I wasn't smart enough to understand it, but I asked, you talked about maturity in the ocean. I asked the question, what triggers these fish to come in at different times of any given age group? Is it maturity in the ocean because of food supplies, genetic variability, or is there some signal that can come to those fish as far as river conditions that encourage them not to mature and start developing eggs and sperm sacs and move up, you know, into the final stages of their life? Do we have any idea what causes this variability? I mean, you touched on maturity, but can you go a little further?
We do. It's a factor that we've studied extensively. And you touched on a couple of factors that do make a big difference. Genetics is very important. Different stocks have different average ages at maturity. Chinook in general have multiple ages at which they mature, and the proportion that mature at each age is an inherited trait. It has variability to it, so it's not always the same. The thing that does influence it, as you had suggested, is growth rate. The fish will respond to a difference in growth between seasons, and that a more rapid growth would tend to cause them to mature early. A slower growth would cause them to mature late. But if you took separate stocks and gave them the exact same environment, they would have different maturity schedules, and the time that they leave the ocean would be also an inherited trait. They're not detecting a signal in the ocean that says it's time to go home. They're detecting a inherited trait that signals the time to go home.
So they may be triggered to go up under less than optimal conditions, and that's just the way it goes.
That is, yes, that's true, and in fact, that really is the challenge the fish face and that natural selection operates upon. It's going to sustain a genetic, since it is a inherited trait, timing of migration. That timing represents an average that works pretty good, but it has variability to it specifically because in some years, a late time of entry would be a big advantage. In a different circumstance, an early time of entry would be a big advantage. And so you sustain in the population a variability around a mean timing. But generally, a specific river system would have a mean timing that works best.
But part of the success or failure is just luck of the draw.
Yeah.
But it would also be, you would want wide variability. You would
Yes.
want some to come in early and some to come in late so that you can have as wide a variety of genetic
Yes, you want that
stock.
diversity, and in fact, it's a really key part of the challenge you face, and one of the things I'm going to suggest in a analytical solution about how you approach finding a balance is recognize that there's a whole lot of variables that we cannot control, and for that very reason, some variability in genetic adaptation and in physical circumstances is useful. It also, even in freshwater, is useful for sustaining fish readiness for saltwater. So there's some relationship. Diversity is a necessary adaptation to a world that we can't predict perfectly for the future. Okay, I'm going to take it to the outer ring. In the outer ring, now we add in all those factors we have management control over that also have can have a dramatic effect on the fish. First of all, at the top here we see we've got these managed effects, and one of them that we've already dealt with and was a major factor in causing the collapse of the winter Chinook run was the fish passage at Red Bluff Diversion Dam. And it was greatly impairing passage, so actions were taken to open the gates and allow for better passage. That made a dramatic influence in those runs starting to come back. Another factor that is very much on the plate still is temperature. Egg survival in that upper basin where the fish are spawning right close to the Shasta Dam and below. Temperature is a critical factor. In the low flow years, it historically killed very large numbers of eggs and still is capable of doing that in low flow circumstances today. So there is a challenge before you. How to balance, we've got to save some cold water so we don't kill the eggs, and you want to use some water. How do you not use too much so you can use it for benefit downstream, but at the same time making sure you're protected for temperature, your cold temperature stores in the reservoir. And just there's other factors too. In this case, you have more variable survival, but we do influence the operations throughout the river and in the delta. We very much control our harvesting. We control hatchery inputs, and there was supposed to be a line in here, but it's kind of missing. But there's, oh, I see. It's down here. Hatchery inputs are certainly another factor. All of those contribute to the whole, and for you to understand what's going on, you need to account for those. And this just is an illustration in the end of, so given a tool like a life cycle model that is spatially and temporally explicit, it enables you then to ratchet through all the cause-effect mechanisms and find out, well, did we use the water resources we've got up here, the stored water, fully to our advantage? There's some losses, some gains. How do we balance those out to a net overall advantage? So just a couple of findings, some science solutions, some thoughts here. Really important to rely on recent studies and best technologies. And I had to smile at the picture of our acoustic tagging operation, a fish getting surgery done, did not look pretty. But what you do need to know is that tests have been conducted to illustrate that those fish survive as well if they're given the proper recovery time. They survive as well as those that we use pit tags on and code wire tags on under the right circumstances. I wouldn't say that we've got it perfectly solved, but we can trust that there are survival differences, particularly if you're comparing one fish to another and you're looking at behaviors. They behave as other fish do. And so a lot of experimental evidence has gone toward testing that. Those recent technologies help you know when and where the fish went. Older technologies only give you a batch answer. You get what 50,000 code wire tagged fish did. You don't know which one went which way. You just know in the end you got some back. So there's new and better data. Second point, beware of correlated explanatory variables. I deal with this up and down the West Coast very often in issues in the Central Valley. Flow and temperature are very correlated. The flow in 1 month is very correlated to a high flow year or a low flow year. So if you choose a month to explain a relationship on, recognize you could have chosen the month before, the month after. We need to establish cause and effect so we're not just playing with correlations that could be spurious. Accumulate factor effects across life stages. Real important to understand where the real causal problem was. Express the effects in adult equivalents. Understand that a fry is not equal to an adult. It takes hundreds of fry to make an adult. And so as you're balancing the act of saving fish, we need to understand what they contribute to the run. And then finally, as I mentioned before, a temporally, that's time, and spatially explicit life cycle model is the best tool for keeping account and then comparing management scenarios. And you will get more on that in a future workshop. Now, just some quick summary points. Scientific findings that support the recommendations. And we've touched on these in Dr. Hansen's presentation and mine here. Majority of natural flow functions cannot be replicated through reservoir releases. You just don't have the capacity to make the same kinds of changes. Large changes in flow produce small and uncertain changes in juvenile survival. Increases in flow may adversely impact reservoir cold water reserves and therefore carryover storage. Salmon survival largely is independent of pumping rates at the CVP and State Water Project facilities in the Delta. Ocean conditions, ocean harvest, and predation are the three typically most important factors influencing survival. It's not every single year, but again and again, those are the key factors that really drive survival. Juvenile salmon mortality of 75% or more upstream of the Delta is high, and that's in comparison to other systems. So some recommended focus for the state board. Protect that cold water pool to maintain suitable temperatures for spawning and rearing. Support the creation, the restoration, and conservation of floodplain and other habitats that would be favored by salmonids. Support the use of non-physical barriers and other mechanisms that might be used to decrease salmonid migration into the interior delta. And that's, pass it back to you, Dave.
Okay, I'm aware that time is passing, so let's move right on. We have our second panel will be on pelagic fish, and specifically Delta smelt and Longfin smelt. Let me just give a quick summary of kind of, instead of at the end, we'll do most of it at the beginning. Life cycle modeling, which is kind of the gold standard for analyzing populations, suggests that the key drivers for Delta smelt are food, temperature, and predation. That nutrients are important drivers of the food web. This is something we discussed at length at the last workshop, and Dr. Dugdale will talk about it today as well. That there's no statistical foundation between X2 and delta smelt, spring, summer, fall, winter. That the low salinity zone and X2 are not surrogates for habitat. They're very inadequate surrogates. That entrainment does not drive delta smelt abundance, and that we've largely solved the problem of entrainment because we pretty much know how to avoid salvage now, at least for the adults. For longfin smelt, we still don't have a mechanism to understand the X2 fall midwater trawl correlation, and even if there were a mechanism, it's a very inefficient tool for increasing longfin abundance. That there are many other factors that are correlated to longfin abundance. This is a point I made last week, and I've brought a bunch of graphs for you to look at today. But that the most important causal mechanisms, or the most plausible in our view, comes back again to food supply and nutrient patterns. And then finally, that we have kind of a plethora of different surveys, and we should be trying to use all of them because they all are adding information that hasn't been fully utilized yet. So just to give an outline of our presentations, we'll start with Dr. Deriso to talk about delta smelt life cycle modeling and delta smelt entrainment. Dr. Noble Hendricks will talk about the statistics underlying the fall X2 and delta smelt analysis. I'll be talking about some of these longfin relationships, outflow, but also other correlations with longfin. And then Dr. Dugdale, Richard Dugdale, will be talking about ammonium inhibition in the food web. I'm just going to quickly give some short bios just so you know who's speaking to you. Dr. Deriso is the chief scientist of the tuna billfish program at the Inter-American Tropical Tuna Commission, and he has been since 1988. This is an international organization that basically manages tuna and other marine resources throughout the entire Eastern Pacific Ocean. He's taught courses in fisheries dynamics for many years at University of Washington, Scripps Institute, and so on. He's been a member of the Ocean Studies Board, which governs the U.S. National Research Council. And he's authored or co-authored over 50 publications. Dr. Noble Hendricks is a consultant. He's affiliate faculty in the School of Fisheries and Aquatic Sciences at the University of Washington. He has more than 15 years of experience as a quantitative ecologist and biometrician, working with estuarine, marine, and aquatic habitats. And he's constructed and implemented experimental designs, analyzed data using linear, nonlinear, time series, and geostatistical models. I introduced myself last week, or last workshop. I've been at Metropolitan for about 10 years and involved in water for about 25 years. And like Chuck, I've been involved in just about everything, but for about the last 6 or 7 years, I've been focusing on ecosystem change in the estuary and trying to understand the population dynamics behind the declines in longfin and delta smelt. I haven't spent much time on salmon. And then finally, we have Dr. Richard Dugdale, who's a senior research scientist at the Romberg-Tiburon Center, which is part of San Francisco State University. And he has a long career in biological oceanography. In particular, he pioneered the concept of nitrate versus ammonium-based food webs in the ocean, and not recently, I'm talking 40 years ago, so maybe he's the oldest of all of us here. He's one of the best-known researchers in the field of phytoplankton ecology in the world and has won many awards from many organizations. And he studied nutrients in the ocean from Peru to the equatorial Pacific with an aim toward understanding how phytoplankton shape food webs and global climate. So I think, except for myself, a pretty distinguished panel. So we'll start with Dr. Deriso from the IATTC. We'll be talking about delta smelt life cycle model results and some of his work on delta smelt entrainment.
Thank you. Good morning. First thing I do is I do the wrong thing.
Yes.
How about this one?
Would you make sure you say whether you're talking about entrainment or salvage? Because yesterday they made a good point of that they're not the same thing.
Okay, I'll try. Thank you. First part of it is I'm going to talk about some life cycle modeling work done with Delta Smelt. The life cycle model is a common tool used to analyze species population declines, well, and increases also. The life cycle model results may provide essential information to future species management actions. On the right side is the first page of a recent publication by Dr. Maunder and myself on life cycle modeling and an application to Delta Smelt. The specifics of the life cycle model that we built is that it represents all the life stages of smelt. We go from larval, well, including early juvenile, into the summer juvenile, and then finally into the fall pre-adult to adult stage. And in between each of these stages, the model models abundance changes between these stages. It allows for multiple factors or covariates to influence survival and stock-recruit relationships. The data set spans 1972 through 2010. The conclusions of that life cycle model analysis was that, first of all, food abundance, temperature, predator abundance, and density dependence are the most critical factors impacting the Delta smelt population. Secondly, entrainment from water export operations is not an important factor impacting smelt population growth rate. Third, that fall X2 is not an important factor impacting smelt population growth rate. And finally, that efforts should be focused on addressing environmental conditions that do affect the species, such as food supply. So as a comparison, there are 3 other recent papers on life cycle modeling applied to Delta smelt. There's a paper by McNally et al., Thompson et al., and Miller et al. And in 3 of the 4 papers, all found predator abundance to be a significant factor. Water temperature in both the Miller and the Maunder Deriso, but then McNally found a summer temperature, so we have that. Prey density was found in our paper as well as in the Miller et al. Other things that were found in those other papers is that the McNally et al. Found the duration of the water temperatures suitable for spawning variable to be important. Also, water clarity, or the inverse of its turbidity, and then winter exports. The factors which were not statistically significant, all 4 of the life cycle models did not find either spring X2 or fall X2 to be statistically significant. In addition, both our paper and the Miller et al. Paper do include entrainment estimates as one of the potential covariates in the model, and neither of those papers found either the juvenile entrainment or the adult entrainment to be statistically significant. And by entrainment here, I mean it in the sense that it's the proportion of the population which is removed by the water diversion facilities, including mortality that occurs while they're in the, like Clifton Court forebay, for example, during that time period as well. The estimates that we used in the Maunder and Deriso for the juvenile and adult entrainment are those that were produced in a paper in 2008 by Kimmerer. And Kimmerer's estimates of entrainment, we used those and then extrapolated backwards for other earlier years that he didn't analyze. His adult entrainment is very well correlated, very much in the way that early results that I had found, namely that there seems to be a threshold level of negative middle river flow, and if the flows are more positive than that, there's no relationship between the entrainment and the old middle river flow. If the flows are more negative than the threshold, then there is a relationship. We fitted that kind of model to the Kimmerer et al. Adult entrainment estimates. The estimates he have fit very well to so then we use that model and its relationship to old middle river flow to estimate what the adult entrainment was in earlier years. The juvenile entrainment, that one, Fish Wildlife Service in their earlier biological opinion found, and I concur with them, that the estimates from Kimmerer's paper are well predicted by a multivariate or multiple linear regression involving spring X2 and old middle river flow. And so I used that multiple regression fitted to Kimmerer's estimates to then plug in estimates for the earlier years. As I said, those two covariates were not statistically significant in the application to delta smelt that we did, but to get an idea of what their effect would be, we did an impact analysis. Now the red line on the top is our predicted fall midwater trawl abundance estimate of delta smelt going back from 1972, and in the publication it goes through 2006. And as I say, the red line does not have entrainment in it. And as you can see, the other covariates, the food, temperature, predation, very well can explain the decline of the delta smelt in the 21st century without invoking entrainment. Just to see the impact of the entrainment in there, we forced the Kimmerer estimates of entrainment into the model, and when we do that, you can see that at most entrainment has had transient effects, that even forcing it with the Kimmerer numbers, generally the pattern that's in that model is very similar to the pattern in a model without entrainment. Okay. I still wanted to understand more about factors that affect. Oh, I did one more, excuse me. This is in reactions, I used a standard life cycle model, in this case it's a Ricker model, to do a steady state forecast into the future of what the population levels would be if we had either no entrainment or a 10% annual entrainment. That is, we either remove 10% of the population on an annual basis or none. And you can see that this one begins in 2011. 2011, as you may know, the fall midwater trawl index showed a big spike, all the way up to 300 and something. That's actually fairly close to the calculated equilibrium for that stock. So the forward projections are coming off of a level of abundance which is already near an equilibrium. And you can see it's a very small effect of the entrainment, for the entrainment. And this is sort of a standard thing that we see in applications in fisheries. I mean, it forms the basis of why we can have a sustainable harvest of fish stocks, is because the populations have these self-regulating mechanisms operating that allow for removals without driving populations extinct, if you will. Then I also wanted to look at management of adult smelt entrainment. The point of this piece of analysis is that turbidity can be used to help manage the entrainment. The data show a historic relationship between turbidity, old middle river flow, and the adult smelt entrainment. So I developed a mathematical model as a function of turbidity and old middle river flow. And the model predicts adult salvage rates when large entrainment events have occurred. So here, to distinguish between the two, so I say the word entrainment, in this section of the talk, think normalized salvage, if you will, okay? Because that is the quantity that I'm working with, is salvage adjusted for populations, adult population size. That is what the data that's used in this piece of analysis. You saw this bubble plot, or something like it, earlier today, and this was the work that I did, and what I've identified on here is the black line, the piecewise linear black line that goes through this cloud of points, and what you'll see is all the little red dots, most of them, 95% of them, lie above the black line. Those little red dots are days in which there was no salvage, okay? And you can see the salvage, normalized salvage is, when there is, it's in blue, and the size of the bubble is proportional to the amount of normalized salvage. And so what you'll also see from visual inspection is that most of the large normalized salvage occurs below the black line. Okay, and when we carry that, to look at daily predictions, you can see that the model predictions in blue, it doesn't get the magnitude very good, but what it does is it locates, to a large extent, the timing of when these large events have occurred. And so I think we have a better understanding now of when you would expect a large normalized salvage and what the two primary factors are that are influencing that. So the conclusions are that entrainment does not appear to affect the delta smelt abundance pattern, and that entrainment levels are related to old middle river flows and turbidity. Thank you.
Thanks for allowing me to present. Just to give a little bit of background, as David had mentioned, I'm a biometrician, and so my focus has been more on sort of the statistical approaches towards answering problems. I also have some work that I've been doing on salmon in the Bay Delta, so I have some familiarity with this environment and in fitting these types of population dynamics to try and understand. Is that better?
Yeah.
Okay, sorry. So we'll go ahead and get started because we don't have a ton of time. So I'd like to also say that this work was done in collaboration with Sheila Green at Westlands, Dr. Brian Manley, who's at West Inc., and Dr. Ken Burnham, who's a retired Department of Interior. And so what we've looked at is fall X2 and delta smelt abundance in the fall midwater trawl. So here's a scatter plot with log of fall midwater X2 along the y-axis and X2 along the x-axis. And if you fit a regression line through this, you'll find that it explains about 8% of the variability. So it's a pretty simple model to define this relationship. And this is often used, obviously, as a first cut for trying to understand how two factors are related. Doesn't include any temporal effects, doesn't include any spatial effects. And so I think partly as a result of that, Dr. Farr et al. In 2011 developed a model to look at how X2 might influence this fall midwater trawl abundance index. And it basically had two steps to their process. The first is that X2 influences the delta smelt habitat index. And then the second is that this habitat index then affects the fall midwater trawl abundance index. So there's two indices that are being constructed there, and then as a result you have this red line, which is that X2 then affects fall midwater trawl index. And so this analysis was what I and my colleagues are looking at. So we'll first start with the first part of the analysis, which is that X2 is related then to the habitat index. And I should note here too that the reason that habitat index is in quotes is because that it's a model-based habitat index. So it's being constructed with a mathematical model, and as a result it has some limitations. We'll try and point some of them out, but so that's why you'll see habitat index throughout here in quotes. So this is this sort of schematic that describes delta outflow. So it's going to describe a physical process, and then it's also describing the manner in which the habitat index was constructed in this analysis. So the physical process is that there's delta outflow that leads to a salinity field, and then that's measured as X2, And that's also measured as conductivity during the time of the fall mid-water trawl abundance surveys. So at the location of the stations where the trawl is conducted, there's a conductivity, and this is electrical conductivity, which is a measure of salinity. So we expect conductivity in X2 to be related because there's this physical process that's tying them together. Okay, on the right-hand side, and so on the right-hand side, we have the construction of this habitat index, which is a function of conductivity, that's the measure of the salinity, Secchi depth, which is the measure of turbidity, and then those are being pulled together in a model to construct a habitat index. And so the point we're making here is that if the salinity field is being used, i.e. conductivity in the habitat index, we're going to expect a relationship with X2. That's the first part of this. So the issue, or potential issue with that approach is that by limiting the model space for the habitat index to basically correspond to conductivity, Secchi depth, and actually temperature was a third covariate that was used in their analysis, that there may be other factors that are missed. So we have a pretty narrow model space that's being used to define the habitat index. So what I'm showing in this table is the results of Dr. Brian Manley's analysis, where he used the information in the Faradol paper to construct a data set that was similar to the data set that they used. He then fit a model, the same type of model. It's called a GAM, which stands for General Additive Model. Basically, it's a very flexible model that can fit nonlinear relationships between the factors and the dependent variable, which in this case is this presence-absence of delta smelt in the trawl catches. So he fit this model, and he used the same one. Top EC is this top electrical conductivity and Secchi depth to the data. And so what we're showing here is the percent of variation that was explained with that model, which is 17.8%. Because conductivity is in the model, the second column shows the correlation then with X2, which is it's high, it's a negative correlation, meaning they have a negative relationship, but this value 0.86 means that they're strongly correlated. In contrast, he also looked at some other covariates that could describe the relationship between the presence-absence and other factors. So and we looked at some static factors. So longitude being a static factor and date being a temporal. So you have basically a spatial factor and a temporal factor. And what he found was that you can explain a little bit more of the variability in these presence-absence data with this model relative to this model. And not surprisingly, the correlation with X2 is slightly lower. Okay. So the question is why? Why do you get a better fit to the data if you're just using these static variables? So I'm going to show you three plots that are going to look very similar to this. And what we have on the y-axis is the proportion of samples that have delta smelt catch. And on the x-axis is the longitude. And since longitude is not the way most of us think of the Bay Delta, we have a couple markers. So we have the middle of Suisun Bay, Sherman Island, and Rio Vista. Okay, so, oops, keep doing that. So this blue line represents this conductivity and Secchi model fit to the observed data. And so we can see with the observed data, there's basically a peak towards the intermediate longitudes with lower values on either end. What we see with the model is that it more or less captures this pattern, except that there's some ranges of longitude where it does, what we call it, there's a biased fit. So what we mean by that is that it's over-predicting over this range of longitude. That means that the blue line is always above the data points. Likewise, throughout the central portion of the longitude, it's under-predicting, so the blue line is consistently underneath the red data points. And at the higher longitudes, or more eastward section, it's consistently over-predicting again. So what we have is this habitat index that's not doing a very good job of predicting the observed habitat over portions of the geographic distribution of delta smelt. If we compare that now with this model that has longitude added to it, we can see that the fit's better over these different longitudinal areas, not surprising, it's got longitude in the model, so that can be used to help factor this relationship. Okay, so at this point, what we have is basically a competing model that's doing a slightly better job of fitting statistically. We're not saying that the habitat needs to be defined by longitude and date. What we are saying is that the model space for defining a habitat index needs to be broadened. And so here's a quote from the EPA technical workshop summary, which I'm not going to read. I am going to point out the fact that habitat really is this large range of salinities, and that the numbers of factors that we should be considering within a habitat index, and therefore within a habitat index model, includes quite a few different components: turbidity, food supply, connectivity, geometry, and, as we just showed, these spatial and temporal components that are inherent in the way that the delta smelt presence-absence is apparent. Okay, now I'm going to look at the second linkage. And in this graphic, we start with the construction of the two habitat indices. So fall mid-water trawl are used as catch data. To create the abundance index, the number of delta smelt in the catch are then used, and there's some calculations made in order to summarize across months and weight those catches among months and among areas to come up with a fall mid-water trawl abundance index. On the left-hand side, we have the same catch data, which we then pull out the presence-absence portion of that. We run the GAM model. We come up with a modeled probability of a presence or absence. Then we do these same index or similar index type calculations, and we come up with a habitat index. So the point here is that we're always going back, and it's all starting from the same data set. So the signal in these data will ultimately get filtered through, and you'll have a correlation between these two, between the abundance index and the habitat index, because you use the same data. So Dr. Burnham ran a simulation study to essentially repeat this type of process where you would generate catches, you would develop abundance information, develop an abundance index, do the same thing with presence-absence, develop a habitat index. What he found was that the correlation you could expect was on the order of 50% to 90%. So if we put that into context with the analysis using the top EC and SECI, the Faradol, that was on the order of 0.51. So we have, which is at the lower end of this range. So it indicates that a lot of this correlation that they're finding in their paper could potentially be due to this process of using the same catch data. Okay.
Were you bothered yesterday? Were you here yesterday?
Yeah.
Okay, by the last presenter from he was brought in by the Sac Valley folks, that the trawl data is not very good, or has a lot of gaps. I guess that was his main point.
Yeah, I missed the last presentation yesterday. You called me out, so. But yes, that's mine. I'm not the most familiar with the trawl data. However, my understanding after looking and working with some of these analysis is that, yes, there are improvements that could be made to the trawl data. I guess I'll leave it at that. Yep.
Would it affect your analysis?
Well, the approach that Brian Manley took to construct these comparisons tried to deal with the fact that there are gaps, that there are certain years where you don't have full coverage, and to account for those appropriately so you don't bias the estimates of abundance, essentially. So he's tried to make post hoc corrections to deal with the fact that you don't have a complete coverage, a data set that's completely covering all times and stations. So.
But I mean, I think it would be fair to say that the central point, which is if you use the same data set on both sides of a correlation, you're going to get a correlation. I think remains no matter what the state of the trawl data.
Yeah. So this is basically just a summary slide, and in the interest of time, I think we can move on from there. What I will say is, so Fish and Wildlife Service made a statement, and this is in a workshop that they produced, and I'll let you guys read this, but basically it's saying that we realize that there is this induced correlation. It's inherently based on a circular argument because the catch was used to define the habitat suitability. So I think there's broad recognition that this circularity of using the same catch data to do two indices and then comparing the indices is problematic. I'll leave it at that. Okay, so here's a couple conclusions. Circularity means that comparing the habitat index to the fall midwater trawl abundance will be meaningless. Essentially, the habitat index is essentially being compared to itself. So by circularity, what we mean is A is being correlated to B, and then B is being correlated to A, and we basically are going around in a circle, when in reality A is essentially B. So doing the correlation doesn't really make a whole lot of sense. I use the term meaningless here, and it's a strong word in my vocabulary, but what I'm indicating is that oftentimes as statisticians or as scientists, when we do a correlation study and we receive a high correlation, we tend to feel that that provides some new information about the relationship between these two entities that we're looking at, or these two quantities. In this case, because they're the same fundamental quantity, when we do the correlation and we get a high correlation value, it doesn't mean anything. It's just comparing the same data to itself. So that's what I mean by meaningless. We're essentially we're not learning anything new by doing the correlation. The second point is that the habitat index needs to, if we're going to build a model that's going to reflect habitat index, that really it needs to have a much broader set of covariates or factors incorporated into it, so that it's so that it can do a better job of reflecting the true or the observed habitat relationship. Thanks.
Thank you, Noble. Let me move on to this very quickly. I'll try to be very brief here. I'm going to actually cut out some of my best slides, which really hurts big time. This is kind of a classic correlation that you've actually seen before, and it's frequently shown. There are a couple things about this. This is longfin abundance on a log scale against X2. And this is like the premier, flow abundance correlation. A couple of things about this to note. One is there's definitely something there, okay? Unlike with Smelt, where I see clouds of points, I see a pattern here. Everyone sees a pattern here, okay? So that's number one. But number two, you see that the line of best fit is dropping year by year, or at least decade by decade. The top line there is kind of the pre-clam, the pre-87 relationship with longfin. The yellow line, I don't know if the colors quite work out here, but the yellow line is basically after the clam but before the pod. And then the red line is the pod years. And you can see that it just keeps dropping. Now the classic analysis of this line is, yes, it's still dropping, but look, the slope is exactly the same as it ever was. So if you put, if you move X2 downstream, you'll get the same fraction fish. You'll get 10% more fish. And that, if X2 is causally related, or I should say if reservoir releases are causally related to increased abundance, that's the case. However, it's also instructive, I think, to take the logs out of this scale and look at the actual linear scales, where the y-axis here now is the fall midwater trawl of longfin. And you can see it used to get up into the tens of thousands. I mean, it was a big number back in the early days. Then the red line here is the same middle graph. That's the late 80s into about 2002. And then the green line here is the pelagic organism decline years. And the point I want to make is that, yes, the log slope is identical. However, this is basically, when people talk about increasing flow to increase population, what they're arguing is that we should be pushing outflow along this green line here, okay? And my argument would be that, yeah, even if it's causal, you can do that, but you're not going to get very many fish, and you're certainly not going to restore populations to anything like what we saw even 10 or 20 years ago. And that from a management and regulatory point of view, a more productive approach would be to try to reestablish some of these earlier lines that have a higher slope, then we're talking about getting a lot more longfin. You could use all the entire unimpaired flow of California and still wouldn't be getting longfin at levels that were seen in relatively dry years, 10 or 15 years ago. So it's really a difficult task to push water out to get more longfin, and it's getting worse all the time. This line is getting flatter every time we look at it. Okay, I promised you correlations. We had a discussion last time, and I'm going to give you all kinds of graphs today, but I'll go through them quickly. I mean, the question was asked, well, you know, if there's smoke, there's fire. If we have a correlation with X2, why don't we, you know, it's just like smoking and cancer. You know, we don't necessarily know the causal mechanism, but we're pretty sure there's something there, even, you know, that we can't quite define. And my response to that is that there are many correlations to longfin abundance, and each correlation has its own implicit hypothesis attached to it. And until we find the actual mechanism, we're basically throwing darts and hoping that we hit the right graph. This top one is another version of the same X2 graph I just showed you, broken into two periods, 72 to 87 and 88 to 2011. And it's a very nice line, okay? Here's another one. This is longfin abundance against unimpaired flow, which is effectively precipitation. And the implicit hypothesis here is that there are all kinds of things that happen in wet years. You got all kinds of junk coming in the river off the fields, organics, you got sediment coming in. Who knows what's going on, but there's a whole constellation of things caused when you have big rainy years, and that maybe that's what's causing this. If this is the true correlation, if this is the causal correlation, releasing very high quality, very clear waters out of reservoirs is unlikely to reproduce the same constellation of things that happens in a rainy year. This is an interesting one that I've been following, which is the idea that maybe it's not upstream flow that matters. Maybe it's local flows that matter. Because a lot of, as I'll discuss later, a lot of the longfin are downstream. I mean, San Pablo Bay is really the center of their population rather than kind of the upstream areas. They may well be responding to local stream flows if in fact flow is what's going on. Napa, you've got Sonoma Creek, you've got Petaluma River, you've got Walnut Creek, all kinds of things, and we have good data on the Napa River. It gives you a nice correlation. The top graph here, Secchi depth against abundance. This is perhaps the best of all the correlations. It suggests that in this case that turbidity somehow is involved in longfin abundance, which could either be they're getting eaten when the turbidity is low, or it could be that they're hiding from the nets and going deep in the water and not being caught when turbidity is low, or that they're evading the nets because they can see it coming in clear water. There are a lot of possible explanations for this graph, but it's certainly a very nice correlation. And then finally, water depth, which is one I would love to get into. It's a particular topic I'm interested in. But actually you can see that the system actually has been getting dramatically deeper, and this is partly an answer to your question about turbidity, Mr. Hoppen, because the system has gotten very deep in the last 20 years. And it could well be that the fish are literally swimming lower than the net can cover. The midwater trawl cannot go deeper than about 40 feet. Many stations are now lower than that. So it could be that we're simply missing the fish. And so now the final set of correlations is kind of the theory that we find most plausible, which is food. The top correlation here is longfin abundance against mysid density. This is a kind of shrimp. And it's a very nice correlation. You can see it's basically, I don't know the exact statistics. It's roughly as good as the X2 correlation, but it has a mechanism. Food correlates to fish. Food means fish, and that's pretty well established. And we've also, talked about the relationship to nutrients. We think that, the nutrient correlations are also very interesting, and that not that the nutrients are directly affecting longfin abundance, but they're probably indirectly affecting it, as Dr. Dugdale will discuss. And I'll skip this one. This is the relationship between mysis, which is the shrimp, and nutrients. And then I'll just quickly talk about different indices. This is the classic fall midwater trawl index against time. You've seen this before on the log scale. I wanted to call your attention to a number of other surveys that are out there. These are 2 additional surveys that take longfin data. This is the Bay study, which is a wonderful survey, which was run by the Department of Fish and Game. And I'm really appreciative that they started doing this in 1980 because it gives us a totally different picture of some of these fish. The Bay study actually has 2 trawls. The blue trawl here is a midwater trawl, just like the fall midwater trawl. You're dragging a net through the water, and then you're pulling it up to the boat. The red line here is the otter trawl, which is basically designed to capture crabs. It scrapes along the bottom. It's got some floats to keep it open, but it's scraping the bottom. And you can see that the otter trawl, for example, the annual patterns are similar, but actually it doesn't show a very strong longfin decline, which is interesting. So my basic pitch is that particularly for longfin, we've got a number of surveys, and we need to be more diligent about integrating the data from all of these different surveys because they all have something to tell us. From the Bay study, this is the distribution of 90% of the smelt during their first fall of life. And you can see that, the fall midwater trawl covers maybe the upper half of that distribution. But the Bay study covers the lower half, and it covers the upper half of the distribution. So the Bay study actually covers more of the longfin distribution. Unfortunately, the Bay study doesn't cover the Pacific Ocean, where some of the longfin may be, and it doesn't cover Sassoon Marsh. But it is covering getting better geographic coverage, and it does have 2 nets instead of 1, so we get vertical information as well. Now, this is adults that are basically on the verge of spawning. This is where the highest 90% of catches takes place. And you can see, again, they're scattered all over the place, with a very large number being below the area that's sampled by the fall midwater trawl. So I think, the Bay study is a very valuable survey that we need to be looking at very carefully because it may change our views a little bit about the long-term trajectory of longfin populations. It also may tell us whether they're moving around in space. This is something you've heard about from Dr. Kimmer and others, that they seem to be moving downstream over time and out of the zone of the fall midwater trawl. And this is the depth chart that I was talking about. The system is getting deeper. They may be literally moving below the nets of the fall midwater trawl. So I'm going to skip these key points. These are the exact same points I gave at the beginning. And so then I'll move on to Dr. Dugdale. And if you'd like, I'll take care of the slides for you. Oh, okay. There you go. Just use the scroll key then. And turn his mic on, please.
Okay, I have inconveniently come down with a head cold in the last day, and so you may have suffered through a few sneezes and snorts and things like that. We'll do the best we can. So this is a paper. What I'm going to tell you today is a quick summary of some of the information that's in this just published paper, but the key points up here are, let's see, how do I get, there we are, okay. Okay, so the key points up here is that this work relates river flow and ammonium discharge and how that may affect spring blooms. So can you hear me? Okay, so use the scroll in